All right, so if this will just cooperate with me, that would be great. So this is just a continuation of the nervous system video. I tried to post part 2 and it didn't upload to YouTube because the file was, I don't know, maybe too big. So I'm trying this again in smaller chunks so that I can actually upload it. So where we left off, myelination, I was talking about oligodendrocytes, which you can see here. And myelination is basically the equivalent of insulation on like the wires you'd find in your house. So the reason that we insulate wires that we find in our house is A, so they don't shock us, but what that represents is really not allowing the electrons flowing through the wires to escape and be able to electrocute us or interfere with other electronics. So myelination is the same kind of thing. So in the central nervous system, the myelinating cells are called oligodendrocytes, which I discussed. Oligo means many, so they're called oligodendrocytes because they have many extensions and projections that they wrap around an axon. In the peripheral nervous system, there's also myelin, but in this case, the myelin is not coming from oligodendrocytes, it's coming from what are called Schwann cells. Now, one major difference, if we look, is that one oligodendrocyte may myelinate more than one axon. So here it's myelinating this one and also reaching across and myelinating this one. Schwann cells, which are limited to the peripheral nervous system, just as oligodendrocytes are limited to the central nervous system, the Schwann cell is going to wrap its little body around the axon, myelinating it, and that means that there is only one axon that this Schwann cell is going to myelinate. It's not going to myelinate any other ones. So what you get is this appearance where there's multiple Schwann cells myelinating this one axon, kind of like beads on a string. So Schwann cells myelinate nearly every axon in the peripheral nervous system. And when we say nearly all or most all, what we mean is not that some of them are myelinated by another kind of cell, but rather that some of them are not myelinated at all. So while it seems like it might be important for every single neuron to be myelinated, the fact is that they're not all, and there are some specific cases and locations where they're not myelinated for a reason. So we'll get to those a little bit later in the term. One thing that Schwann cells do is they participate in the repair of axons that have been damaged. So while neurons are not mitotic, they don't reproduce by mitosis any longer, they are capable of fixing an axon that has been cut off. It takes a while, but the Schwann cells can help the axon find its way back to where it used to go. So, even in unmyelinated axons, you will still find them associated with a Schwann cell. It's just that the relationship between the Schwann cell and the axons does not look the same in myelinated versus unmyelinated. So this is what that picture or this figure is showing you. So if we first go here, what you can see is what the Schwann cell is going to do is just wrap its membrane many, many, many times around the axon like a little jelly roll. And then if you take a transmission electron micrograph of that, which is a way of looking at super duper tiny stuff microscopically, you can see that there's a very obvious layered wrapping. If we go over here, what we can see is that in unmyelinated axons, although there's still a Schwann cell, it's not doing that jelly roll-like wrapping, it's just kind of giving them a hug. So it's partially insulating them from their surroundings, but not in this robust multiple layers wrapping way that myelin is. And the significance of that is that the electrical insulation function that is performed by myelin is not performed in these unmyelinated axons, even though they're associated with a Schwann cell. So, sorry, I had to pause there for a moment. So as you can see, Schwann cells are involved in both, let's move this over here, myelination, where they wrap around and around, and in unmyelinated axons, the take-home message there is really that regardless of whether an axon is myelinated or not, it needs to be protected, at least somehow. So Schwann cells do both. And I want to take a moment to point out this word here because it's a little bit confusing. lemma means membrane, and neurolemma would suggest, based on the other lemmas you've experienced so far, that it was the cell membrane of a neuron. But in this case, it's not. It's the membrane that surrounds and encloses unmyelinated axons. So it's just a bit of a misleading name, which is why I wanted to highlight it here. Because as you can see, if we go over here, come on, there we go. This is the axolemma, which is pointing to the cell membrane of the axon itself. So just a little vocabulary hint there. Neurolemma in this case does not actually refer to neurons. It refers to the Schwann cell membrane that encloses unmyelinated axons. Okay, so that was Schwann cells and myelinating, or unmyelinating, as the case may be. The other main kind of peripheral nervous system glial cell are called satellite cells. And these tend to sort of cluster and surround a neurosoma, so the cell body of a neuron, and those are in ganglia. So let's take a moment to talk about what a ganglion is. So ganglia is plural, ganglion is singular. And let me actually just shift my computer into drawing mode here, now that it's charged up enough, so that I can sort of, oopsie, wow, that was exciting, explain what I mean when I say ganglion. So I think it's worth noting. So this is the singular form, and this is plural. So the definition of these is a group of neurosomata in the peripheral nervous system. So instead of having a bunch of neuronal cell bodies located just kind of haphazardly wherever, when there are a bunch of neurons that are gathering information to or from somewhere, or they're clustered somewhere in the peripheral nervous system, as they often are clustered together, when a bunch of them are in a group, we call them a ganglion. And then within the ganglion, each of the neurosomata, bam, has its little sort of shroud of satellite cells around it. And they do things like protect the neurosoma. They also help to regulate the gas exchange coming into, in the case of oxygen, and leaving, in the case of CO2, the neuron as the neuron respires. They can provide nutrient sources for these very, very nutrient-hungry cells, the neurons. And they're also involved in manufacturing and recycling of neurotransmitters. So make and recycle. So continuing with our theme that we were talking about in the central nervous system, and just about cells in general, if we're going to have cells that are really, really busy, like neurons, doing lots and lots of things, so biosynthetic activities, depolarization activities, if you're going to have those cells, they're going to need a support staff of some kind. So even though neurons are sort of the star of the show, as far as getting the most attention for neurophysiology and all that stuff, this is why we have neurologists, not gliologists, as hospital specialists. However, none of the work of neurons really gets done without neuroglia. So while they are not necessarily the central stars of the nervous system, they really shouldn't be ignored because they have a huge role to play. Okay, so I would just like this to go. Sorry, 1 second. I'm going to pause this because my controls for PowerPoint. Okay, so I got my controls back. It was just I was a little too zoomed in, and since I'm in tablet mode, I didn't have my option to do escape key. So let's have a peek, oopsie, at neural responses to injuries. So I mentioned earlier that neurons are amitotic, meaning they don't reproduce by mitosis. But that doesn't mean they're incapable of healing from injuries. So let's say you are cutting vegetables on a cutting board, for example, and you accidentally, whoopsie daisy, the knife slips And you end up severing one of the nerves in your hand, which, ouchie, A, but B, you're going to need that back because neurons are how you control your hand muscles. Fortunately, depending on where the injury occurred, you can actually regrow axons that go back to where they were, and Schwann cells play a role in it. So there's a reason that this only occurs in the peripheral nervous system, and that is because the organization of the peripheral nervous system is a little less chaotic than the central nervous system, where in the PNS you have bundles of myelinated axons, which we call nerves. And they're generally running in the same direction as each other along a line. So there's, within a nerve, there's not a whole lot of interconnectedness between and among the axons of that nerve, which makes healing and directional healing specifically a lot more easy. With the central nervous system, there's too many axons, and there's also axons that cross past each other and intersect. Astrocytes also produce really dense scar tissue, which is impossible to grow through. And finally, astrocytes also secrete various inhibiting factors that help to stabilize your central nervous system and how it's wired up by making sure that the neurons don't change their mind about what synapses they want to participate in to a large extent. Now, that's not 100% the case, because the more we understand about the brain, the more we now know that adult neural remodeling is much more common than it used to be thought of as, but it doesn't reach the extent of being able to do robust central nervous system damage healing after, for example, an axon injury. So while we've got it up on this slide, let's have a look. And I'm just going to go ahead and move this over here. So what you're going to see is at the site of the injury. So I gave the example of we, cut ourselves with a chef's knife. And now we're just down one axon, probably to some finger muscle or something like that. So there's our little knife. Knife skills are important, people. A falling knife has no handle. It's one of the first things you learn in a kitchen. So, as you can see, there's this stump here. So we have a proximal and a distal stump, meaning which one is nearest to the cell body, or soma. So the proximal stump is the one that's capable of growing outward. The distal stump, because it's not connected to the neurosoma, is not capable of that, and that ends up getting cleaned up by our friends, macrophages. So the Schwann cells that used to myelinate the distal stump, they retract and then regroup, and they begin to undergo mitosis. And what they're going to do is, as you can see, there's 1, 2, 3, 4. They're undergoing mitosis, and they're also sticking to each other. And with each mitotic event, they stick further out towards the proximal stump, and it makes a little road that the proximal stump can follow. So the little Schwann cells are saying, hey, look, we made you a little path, and they're also secreting a bunch of cell signaling factors to attract the growing end of the axon down the little path that they're creating. So as it continues to grow and advance, the Schwann cells get back to their work of myelinating by wrapping around and around and around until the axon stump regrows, and it regrows to its original location. So it's really cool that we can do this in the peripheral nervous system, which means that over time, if this is, say, a motor neuron to your index finger, that means that you can get motor control of your index finger back, even if you sliced up your nerve that's controlling it. So the other thing to consider about this, the factor that is not explicitly mentioned here, but is important because it's relevant clinically, is that this is not a fast process. It takes a long time. So, because of the nutritional demands of neurons and because they are slow to grow and heal, if you get a neural injury, although healing is possible, depending on where you have the injury, it's going to take a long time to heal. So that's just helping manage expectations about what's possible with nervous system injury and recovery. Okay, so this next portion of the PowerPoint actually covers stuff that I also talk about in the graded potential, resting potential, action potential videos that were more Khan Academy style that I drew for you. So I'm going to talk through what's on these slides here, but if you want a really detailed rundown of each of these things, so the one about the resting membrane potential, I talk about how that's set up. So with sodium potassium pumps and leak channels, etc. We also talk about graded potentials in another video where we talk about chemically gated ion channels and other things. And then we have one that's talking specifically about the action potential, etc., so forth. So in addition to what I will talk about right now, you also have those resources and the podcast episode I made about the same content. So this is a topic or a set of ideas and concepts that are not going to be necessarily intuitive for the majority of students, because if you're not used to thinking this tiny and you're not used to thinking of electricity on terms of ion movements, it can be difficult to wrap your head around. So all that is just to say, I'm offering you lots of different ways to consume this content. And if you do all that and you still struggle with it and we need to talk about it in class, that is fine. I will draw it out on the board or do whatever it takes to help you get the rest of the way there. So with that, let's begin, shall we? So as I mentioned in the other videos I made, and in the I emphasize this in the podcast especially much, we're going to focus on potential. And that word, that piece of terminology comes up a lot as well. So if you're like, potential what? Potential energy. Or potential to do work. So in physics land, doing work is just a way to talk about substantive change in a system from a resting state. So that's what work means. So if the goal of a neuron is to transmit information in the form of electrical signals, then that represents work, right? So if I'm going to make electrical signal go from place A to place B and move, that is a change of a system, and that is doing work. So according to the laws of the universe and thermodynamics, if you want to do work of any kind, it necessitates an energy conversion step where you take energy in one form and you turn it into another form. So if you're having a hard time wrapping your head around that, and you're thinking in abstract physics terms, or you're like, I haven't even taken physics, I don't know what you're talking about. Don't worry, there's lots of work that you've seen done physically in your lives that is more intuitive probably to think about just because you're used to the things I'm about to describe. So let me think of some examples that might be more familiar to you. For what I was saying was for doing work, I marked up the pause button with my pen and I clicked it because I was trying to erase the mark, but I will just leave it alone now. So examples of doing work. Let's say you want to, play a tape. And I know that's a dated reference because we don't really use tapes anymore, but most people have a passing familiarity with the idea that if you want to play a tape or maybe a vinyl record, it's got to spin around, right? That's kind of the central idea for tapes, is there's spinning happening, yes? So if I want the spinning to happen, let's say it's a battery-operated thingamajig. And so if I want to make the rotation that makes the media play occur, I have to put batteries in the player of some kind. So batteries are chemical potential energy that is stored up in a little package. When you separate chemicals in a certain way, it represents potential energy that can be used to do work. So when you put batteries in the little player, and then you flip it on, the energy coming out of the batteries is the separated chemicals becoming less separated from each other, and that releases electrons that are able to then turn the potential energy from the battery into kinetic energy, which is the energy of movement. That's rotation. And the rotation is picked up by a sensor and turned back into electricity to travel to your ears, either via headphones or a speaker. Speaking even more simply, let's say you want to make a fire because you're cold and you don't have a lighter. If you think of ways that you could make a fire, here's one. You could take one flat piece of wood and then make a little dent in it, and take another cylindrical piece of wood and then swiftly rotate the cylindrical piece of wood into the flat piece of wood using either your hands or a bow drill. Look up bow drill fire if you're curious. But if you do that, you are creating friction between the two pieces of wood. You're doing that, you're creating that friction by turning the kinetic energy of movement, by spinning that piece of wood, and force. You're turning that into heat energy to heat up the frictive surface between the two pieces of wood, which eventually causes a combustion reaction to start, and that's how you get an ember. And then you can take that ember and you can set it gently, gently, gently in some easily burnable fuel. And the combustion reaction that you started by turning the kinetic energy of movement into the friction heat energy that you use to start the fire, you can keep feeding more and more fuel into that. And as the fire grows, it's going to use oxygen and the carbon-based fuel you give it to continue doing that work. But the initial start was you taking the energy of rotation and turning it into heat energy. You did that, if you can do it. So those are some ideas around potential energy and changing energy formats that might seem a little bit more familiar to you. So transmembrane is the other part of this word, and trans means across, and membrane, you guys know what a membrane is, that's just a membrane. So if I want to have potential energy that is across a membrane, typically what that looks like is charge separation and chemical separation as well. So for a moment, I'm going to blank out the screen here. And we're just going to look at a basic idea. Oopsie. And that idea is just going back to some really basic ideas about diffusion that you probably haven't thought much about since Biology 160, but it's useful to revisit them a little bit here. So I'm just going to draw myself a membrane real quick. I've drawn the little yellow lines represent the phospholipid heads, which are hydrophilic, and these orange guys I'm drawing are the fatty acid tails of the phospholipids, which are hydrophobic. So let's just go ahead and label that hydrophobic zone. So the reason I'm labeling it is because the hydrophobic zone and a compound's inability to cross it is what helps keep this membrane having two sides, one side that is out and one side that is in. So if I have the same number of things on each side of the membrane, so let's just say, for the sake of argument, I'm going to just do blue pluses, doesn't matter what they are. So if I have my blue pluses, and you're thinking about what you learned about diffusion from Biology 160, and you note that there are 1, 2, 3, 4, 5, 6, 7 pluses here, and only 1 here. If you remember that the laws of diffusion state that particles want to go from an area of high concentration to an area of low concentration, and then I said, okay, so remember that and then tell me which way the pluses are going to want to go. The majority of you would say, well, they're going to want to go this way because there's more of them outside of the cell and fewer of them in. And if I then asked you, okay, so when do you think they would stop moving? You would probably tell me that they stop crossing the cell membrane when there's an equal number of pluses on each side of the membrane. So now that I have 4 pluses here and 4 pluses here, there's not going to be any net movement of pluses across the membrane because we have reached equilibrium. So now there's no net movement, meaning we're not going to do any more work. So if we know that equilibrium is lowest energy state and represents very little in the way of potential energy to do work, the opposite must also be true, where if we have an extremely polarized membrane that's not in equilibrium or even anywhere close, so let's just add a bunch of pluses out here. And I have no pluses on the inside of my cell. So if you know that the steepness of a concentration gradient is one of the things that powers diffusion, and if I were to ask you based on this picture, okay, how badly do you think those pluses want to cross the membrane? You would probably say, pretty badly. They really want to go across to equilibrium, especially since the concentration gradient is so steep, where there's 0 here and lots out here. That represents a lot of potential energy because the potential is for the pluses to diffuse. So if I'm not letting them, I'm saying, oh no, you have to be out here, you can't diffuse across. This represents stored energy, and specifically potential stored energy. So that's what I mean by energy conversion and what I mean by potential energy. So if you want to create a situation where you can do work in a cellular level, but you're not doing it currently, a great way to do that is to make 2 sides of a cell membrane have different things on them and in different amounts. So if that seemed reductive to you, I apologize, but I promise I'm coming to a point. And that point is the following. Here, I have a situation where we can look at this and make some of the following observations. 1, if I look at how many sodium ions are outside, and I guess let's just make them be purple since that's the color the book is using. I usually make sodium be green. So sodium here is this purple color. So I think if you count, the outside of the cell has lots of sodium, and the inside of the cell has very little sodium. If I count the opposite thing, and I'm saying, there's lots of potassium inside of the cell, and very little potassium outside of the cell, we have a charge separation and a chemical separation here, where the concentration of these 2 chemicals is the opposite on either side of the cell membrane. And also, if we go ahead and just look at the number of pluses and minuses, minus, 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 these large proteins are mostly negatively charged. And then I have a handful of chloride ions as well poking around here. That's a lot more negative charge on the inside than the 1, 2, 3, 4, 5. 5 units of negative charge out here. I'm not even going to count them here. Probably around 20 units of negative charge in here. So we've got a situation where 2 chemicals are separated from each other in different proportions. So we call it a chemical potential. And then we also have this difference in charge. So this is an electrical potential. Electrical referring to electrons. Remember, a single electron is a unit of negative charge. So all of these negative charges represent an atom or a compound that has basically missing a proton and therefore has one extra electron. So the other way to refer to the transmembrane potential, according to that, is also the electrochemical potential. Electro referring to the electrons, and chemical referring to the chemicals. So it just so happens that if you measure a neuron and you take into account both the number of pluses and minuses on either side and which ions are carrying those pluses and minuses, if you measure the inside, which is what this is showing you, it's a probe that's measuring the number of charges inside, and then there's a reference electrode out here, which is measuring the positive charges out here. What this sensor is doing is just saying, hey, if I add up all of the charges on the outside of the membrane, and I add up all of the charges on the inside of the membrane, how different are the two sides? So it just so happens that most neurons sit at a condition where there's about -70 millivolts difference, meaning that the inside of the cell is -70 millivolts more negative than the outside. So negative here just means that there's more negative charge and less positive charge inside the cell. So this -70 is our reference resting membrane potential for a reference neuron. So in reality, many neurons have kind of slightly different resting potentials depending on the cell, but if you sort of ballpark them as a group, they tend to shake out to about -70 overall. So that's the reference number that we're going to use. So the ion concentration obviously differs in the extracellular fluid compared to the cytosol. That's the concept I just built up. And then the other thing that I haven't quite addressed yet is, okay, so how do you get the cell to a place where it has that membrane potential? Well, we have to remember that cells are selectively permeable, meaning that they do let things across, but only some things. And the cell also controls in which direction it crosses. So some things can only get out and not in, other things can only get in and not out, some things can go either way, and the cell controls this using transmembrane proteins. And these proteins can either be proteins that are going to do work, so things like the sodium-potassium exchange pump, or proteins that basically just create an open tunnel for stuff to pass through. So let's just have a look at the sodium-potassium pump. So what it's going to do is spend an ATP. So this takes energy. So part of your daily calorie budget where you eat food and you turn that food into ATP, part of that is spent here, where you're going to cleave that last phosphate off of the ATP, and that's going to allow you to kick 3 sodium out of the neuron and gather up 2 potassium. So if you look, you're losing 3 units of positive charge, you're only getting 2 back. So the net loss of positive charge every time this protein does its job is -1. And if you have lots and lots of these, which neurons do, all spending ATP to do this same step, you can get to a very negative membrane potential relatively quickly. So, leak channels make up the other part of the equation. So again, I'm going to black out the screen here to just do a little thought experiment with you, and that is, let's imagine let's say we have a membrane that is at, I don't know. Here's negative, I need this one, there we go. -70, what about here is +30. And let's ignore all the other channel types for now, but we're going to say that this cell, for whatever reason, is up here at about +10 millivolts. Don't know why, doesn't matter why. If I, at time 0, turn on all sodium potassium pumps. And you know that every time the sodium potassium pump spends an ATP, the cell loses 1 unit of positive charge. You would expect, after I turn on the sodium potassium pumps, our membrane potential to start dropping more and more into the negative. So the question is then, if we're using only the sodium potassium pumps, why doesn't it just keep dropping? Because if this pump has just got an endless supply of ATP and it's always kicking 3 sodium out and letting 2 potassium in, always losing 1 unit of positive charge per cycle, why doesn't it just plummet forever? How does it reach resting? And the answer is leak channels. So if I'm going to erase a little bit of this here. So if I let some sodium and potassium leak back down their gradients, at some point, depending on how many leak channels I have in the membrane, at some point it's going to reach the point where the amount of sodium and potassium leaking is equal to the amount being turned over by these pumps. And that's how we get to flat. So at this inflection point, I'm going to say, okay, so not only do we turn on all of our sodium and potassium pumps here, but at this point we're going to say, turn on all leak channels, which are going to oppose the charge separation that the sodium and potassium pump is doing. Not perfectly, because remember, we're still at -70, but we've let the sodium and potassium pump get down to -70 before we decided to make the membrane be leaky. And now we have a situation where we've stabilized it at -70 millivolts. So that's the idea behind leak channels. If you don't want the membrane potential to keep going down and down and down and down, which we don't, and we want to stabilize it like we have here. So this is a big green check. We want that. We do not want this. We need leak channels to just let stuff leak across and oppose the action of the sodium and potassium pump. So that's the idea behind what is going on here. So the leak channels are always open, and I'm going to highlight this as being even during an action potential. So it's not like they turn off during an action potential. There's also a non-equal number of leak channels. So we have more potassium ones than sodium ones, which means that potassium is going to leak out more so than sodium leaks in. We also want to note that there's these large proteins, which are things like enzymes and transporters inside of the cell. And because amino acids, many of the amino acids in the 20 or so amino acid chart that we have are negatively charged. So if we know that a lot of the amino acids that make up proteins are negatively charged, and we know that proteins are long chains of amino acids, then it would stand to reason that large proteins are going to be mostly negatively charged. So they contribute a lot to the inside of the cell's negative potential, and they're too big and too polar to get through the hydrophobic middle layer of the cell membrane. So they don't, and they just stay inside and contribute to the negative charge on the inside. Now, there's one additional factor here that we have to consider, and this is another membrane problem. So I'm just going to draw it over here. So I'm going to draw a very rudimentary membrane, pretend like it has all of these parts, but I'm just going to make it very simple. So we know that opposites attract, right? So if I have a bunch of negative charges here on the inside of the cell, the reason they're right up against the membrane is because on the outside of the cell, where there's more positive charge, that's where the negative charge's girlfriend is. I know that's anthropomorphic and silly, but it's true. So the negative charge is like I long for you. It really wants to be near the positive charge, but it can't get there because the membrane's in the way. And the same way that the positive charge, because opposites attract, is like, I long for you too, minus sign, but we can't get to each other. And so, instead, they just, like people who are in love on either side of some kind of barrier, they're just gonna stay right by the barrier, looking at each other and longing for each other, but unable to reach each other. And so this makes the area specifically near the cell membrane extra specially separated, with a lot of potential energy compared with further away in the extracellular fluid or deeper into the cell. So that's the final sort of aspect of all this resting membrane potential stuff. So we talked about the sodium-potassium pump already. I just want to do a quick remind that it costs ATP to do this. Oopsie. So if we have the situation where we have 3 pluses out and only 2 pluses in, we have a net loss, which means we have -1 per turn. But we also have leak channels. That ultimately, again, shakes out to -70 millivolts. So what can we do with that -70 millivolts? We'll build that concept up next. And in doing so, we'll also, talk about what those things mean for neuron function and how ambient, read plasma and extracellular fluid, ambient sodium and potassium concentrations make a huge difference. Make a huge difference for neuron function. So this is why hypokalemia or hyperkalemia, so this is potassium illnesses, so hypokalemia is low blood potassium, hyperkalemia is high blood potassium. These both cause neurological dysfunction. And so do the sodium-based problems. So hypernatremia or hyponatremia, which are low or high blood sodium levels. So if basically the idea is, if you mess up the extracellular fluid's composition and ratio of sodium to potassium, let's say, for example, using this picture, we have a situation where we have hyperkalemia, so we have too much blood potassium for whatever reason. And so we have hyperkalemia, which means that our extracellular fluid, which comes out of our blood, is going to be, let's say it's about 100 millimolar instead of the usual 10. And we also have hyponatremia, meaning low blood sodium. And instead of having about 140 millimolar, we're down to about 11 millimolar. So if I have blood potassium that's too high and blood sodium that's too low, and that's going to change the external environment compared to the inside of my neuron, that creates a situation where in both cases, the ions on either side of the membrane are close to equilibrium. And if we're close to equilibrium, that means very low potential to do work. So what you see in this case, or would see in this case, and this is an extreme case I'm just using to make a point here, but if there's almost no potential energy available to do work and you don't see any diffusion, you get the heart muscle stops, neurons don't fire, and that equals death. So we want to avoid that. So differences are key, and the right differences in the right proportions are also key. So as you move through your careers, especially if you're here for nursing, what you're going to notice is that, especially when you get to clinicals during nursing, charting elements like keeping track of your electrolyte balance in your patients, especially if you're doing things like infusing them with various IV bag stuff, you're going to have to keep pretty close track of the correct and appropriate range for those two ions in particular, sodium and potassium, because otherwise you could result in a patient who has got failing cardiac or neurological function, and that's obviously very, very bad. Okay, so we want to create a resting membrane potential, which has the potential to do work. And if I was smart, I would have Work by Rihanna queued up to play briefly here, but I'm not. So you're just going to have to imagine that someone in the background is like, work, 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 work. That's what this is all about, work. All right, so work, which I've spelled wrong on purpose here, equates to change. So we've gone from a place where we've got a situation where there's a charge and chemical separation across our membrane, and that creates the potential for some change to happen. But as long as the change is potential, that means it's not currently occurring. So what happens when we change from potential into actual? So potential meaning possible work, but now we want to do the actual work. So in the case of an electrochemical potential across a membrane, what that actual work looks like is ion movement. And specifically ion movement across the membrane down their concentration gradient. So in a neuron, what that means is we've got sodium going into the cell and or potassium going out of the cell. So they're falling down their concentration gradients. So this brings us to the concept of channels. So a channel is a protein-based passage through a cell membrane. So it's something that opens up and allows charged or large particles through, because again, the charged and large particles, they can't make it through that hydrophobic region in the middle of the cell membrane. So if they're going to make it through, they need assistance. So if you want to make a channel that is dependent, meaning it's open under some circumstances and not others, there's a couple ways to do that. One of them is voltage gated, meaning if the membrane potential changes to their favorite voltage, they will open up, and if it's not at their favorite voltage, they will shut. So gated channels, the beauty of these is they can be turned on, open, or off, shut, according to a variety of stimuli and under a variety of conditions. So some of them are chemically gated, like, for example, the chemically gated ion channels at your neuromuscular junction. Remember, those are acetylcholine based. So these are going to open up when their favorite chemical is bound to them. Voltage gated opens at their favorite voltage range. And mechanically gated are a little bit simpler. They just open up when they're smushed. So you guys might remember the lamellated or Pacinian corpuscles that we learned to recognize in the histology of the first lab stuff. the way those work is they're full of these chemically gated ion channels where if the Pacinian corpuscle is smushed physically, like you're touching it and you're pushing on it, that force opens the the mechanically gated ion channels and creates a receptor potential in the Pacinian corpuscle that activates a sensory neuron. So that's a direct example of something that you've had experience with already in class that works on this principle. But regardless, none of this is possible without a resting potential. So, moving right along. Let's just do a quick review walk through the different kinds of channels that I've just described. Oops, sorry. Because repetition is our friend when we're just learning stuff. So here's a nice little visual of what a chemically gated channel would look like. And this happens to be, oh, look, acetylcholine, which is the neurotransmitter responsible for, among other things, motor control of skeletal muscles. So you might remember acetylcholine from the neuromuscular junction. So in our resting state, we have the channel is shut. So here we have a sodium that wants to go through, but it can't because the door's not open. Now, if we get the introduction of one acetylcholine binding to its binding site, that's great, but it's still not sufficient to open this channel. We need 2 acetylcholine to open this. So the reason I'm emphasizing the fact that 1 isn't enough and we actually need 2 is because that's one of the special things about chemically gated channels that allows them to produce graded potentials, is that if I have a population of these channels all next to each other, so let's say I have 200 acetylcholine receptor ion channels. If I have 200 of these channels, but I only have 100 molecules of neurotransmitter, that means I'm only going to open about half the channels. So what this means is that the amount of chemical, typically a neurotransmitter, although not always, is proportional, which is what this open infinity symbol means, proportional, to the number of channels that are actually able to open. So if I have channels that are letting ions through, and I know that there's a variable number of these channels that can be open at a time, and it's going to depend on the chemical, that means that the amount of ion flux meaning the ions crossing the membrane, the amount of ion flux is also proportional to the number of channels open, and therefore to the amount of chemical. So, if we know that pluses crossing the membrane changes the voltage, and I'm going to let some pluses inside and make my voltage be a little bit higher than my negative 70, chemically gated ion channels are responsible for graded potentials. Because graded means variable magnitude. So that means that you can have different flavors of graded potentials. You can have a big one where there's a lot of change. You can have a small one where there's not very much change. Sometimes the change is enough to get us to action potential territory. Sometimes the change is not. So any gated channel whose amount of openness is contingent upon a chemical is going to produce graded potentials. And if you're like, why are you belaboring this point so much? What is the deal with this? When I explain action potentials, you will understand. So just stay tuned. It's also significant, before I move on, that these channels tend to be found on dendrites and neurostomata, which is the receiving end of a neuron. So the message is neurotransmitter, and when the neurotransmitter binds to receptors, which are often also chemically gated channels, that constitutes receipt of the message. So that is in contrast to what is present channel-wise on the sending part of any neuron. And so again, if this isn't making sense to you right now, why I'm belaboring these points, when we get to voltage-gated channels and where they're located, that will make a lot more sense, because we will want to compare and contrast the two. So before we do that, we will take a brief foray into mechanically gated channels, but not super hard, just because they're not very hard to understand. So like I said, when you squish them, they open, and when you're no longer squishing them, they shut. So I think that's pretty easy to understand here. As long as I have the membrane in its natural configuration where it's not being mechanically or physically distressed in some way, the channel is going to be shut. But if I poke it, like so, let me just draw a giant finger. So here's my giant finger, there's my fingernail. I'm just going to go ahead and poke this. There's my little knuckle. It's just the finger of God is reaching down and pushing on this membrane. Then we get ion flux, and that's what creates a potential. So these are generally mechanically gated channels are one type of receptor potential, which means you're detecting some kind of sensory information, specifically tactile sensory information. So let's put tactile, but also hearing and balance. These are also mechanically gated ion channel thingies. So those are pretty easy to understand just looking at this picture. So let's move on to voltage gated ion channels. So voltage gated are open at their favorite voltage range. So if we look here, this particular voltage gated channel is shut at -70, so it's shut at resting. If something happens where I get the membrane to -60, that's the opening condition. And then notice I have this inactivation gate here where it's going to be open at -70 as well. It's also going to be open at -60, but the moment we get up to +30, that sucker snaps shut. So we have what I like to call the Goldilocks range of voltages, where it's the right combo of voltages or the right range for this very, very fussy particular little channel to be open. And at any other voltages, it is not open. So, graded potentials, let's just draw ourselves a little graph here. This is a graph you're going to become very familiar with. So I'm going to say this is millivoltage, and this is time. And the time is typically expressed in milliseconds because all of this is happening very quickly. So graded potentials, I'm going to say, are orange. So let's say I have, let's mark -70 off, -70, and then this is -60. So if I have some voltage changes where my membrane is like doo-doo-doo, just minding my own business, being at rest, and then something comes along to make the potential change, and it's like, oop, it goes up a little bit, but then it goes back down, and then the membrane potential is back to resting. And then maybe it goes up a little bit more, but it doesn't quite make it to -60, and then we just get a decay back down. So you can see that these two potentials, oop, oop, are at different heights, meaning they don't have all the same amplitude or height of the curve. That's what graded means. They can vary in their intensity. If we get one that goes to -60 though, see there? Because that's our threshold voltage. Then, boop, we're going to go up to +30. and then back down to about -90. So graded potentials, again, are the domain of chemically gated ion channels, or in some cases mechanically, but usually chemically is kind of the main deal. And then -60, what you might have noticed is that, if I can erase this here, oh, -60 is the threshold voltage. So this green part of the curve is when the voltage gated channel snaps open, and then +30 is when it snaps shut, but another voltage gated channel snaps open, which causes the membrane to go back down. So action potentials are caused by voltage gated channel changes, and action potential is this spiky waveform. Graded potentials are these rounded, less spiky waveforms. So that's what I mean when I say graded and action potential. Different waveforms, different shapes, different laws governing them, depending on what the rules for opening and closing the channels are. So let's have a look at a graded potential. So what we're going to do, is just take a peek at this figure from your book. So notice here we have resting. So we know that we've got this resting state where there's a lot more positive charge outside of the cell than inside of the cell. And in this case, we're going to ignore potassium for now. For right now only, we're just looking at sodium. And the reason we're looking at sodium is because it's the most frequent ion that causes action and graded potentials, and also it's the one that's most relevant for neurons. There are other options in other places, but this is the main one, so that's why we're focusing on it. So we've got chemically gated sodium channels on the neurosoma, so the cell body, and you'll notice that their active site is currently empty, so there's nothing bound there, meaning that these channels are closed. Now, if something releases a chemical that opens the sodium channels, and we have one of them open and then another one, that gets us about 5 millivolts more positive. So we've gone from -70 up to -65. Now, here's where some more diffusion rules kick in. Up into, so at time 0, let's do time 0, there was no sodium right here inside of the membrane. Now suddenly at time 1, there's lots of sodium. So if we know that sodium goes from an area of high concentration to an area of low concentration, and we know that there's also no sodium over here, it only makes sense that the sodium, after it enters, it's not going to hover here in a cloud. It's going to go over to the neighboring inside part of the cell where there's no sodium. So it's going to diffuse laterally once it gets in. And that lateral diffusion we call a local current, meaning that the laws of diffusion dictate that once this sodium makes it inside of the cell, it's going to wander over next door to where there isn't any sodium yet, because diffusion tells it to. It's like, well, I gotta go where there's not as much me. And so off I go to this area nearby where there is not as much me. So this local current helps to move and spread the site of excitation across the membrane. And if there's enough of a local current, we might be able to loop voltage-gated channels in at the axon hillock, and that's what would start an action potential that will be able to propagate down the axon. So I know this is tedious conceptually. I'm trying to be intentional about building it up piece by piece because I know that this is really complex information and it's easy to get mired in the details and lost along the way.